Hi there. In this last video about proteomics, we'll talk about how we interpret the raw mass spec data. We'll say a few words about one of the advantages of proteomics, and that's our ability to measure post-translational modifications. We'll talk about protein quantification, and we'll end with a few words about the comparison of protein and mRNA levels. At the end of the last video, we had gotten to the point where we had produced this kind of raw data. We had measured fragments of various m/z units, or various masses, and we've recorded them along with some intensity information. The challenge for us now is to get from a raw uninterpreted spectra to an interpreted spectra where we've applied a molecular interpretation based on known proteins, where we identify the peptide that has produced this pattern. Now, because it's so conceptually appealing, you'll think that the plan is to measure the distance between two peaks, record the mass that it would be, and interpret in this case that it would be a leucine amino acid that is responsible for that, and that would be the way that we would interpret this molecularly. In principle, this is a very valid way to go, but in practice, it's too complicated still. The algorithms, while getting better, do not work well enough that way, and so I want to guard you against thinking that. This slide summarizes how things are done. We have two arms to the process, one above the line and one below the line. The data acquisition part is we have our proteins in our sample. Remember, we digested them with trypsin. That cut them into peptides ending in lysines or arginines. And then we took one of these peptides at a time and generated an experimental spectra in an MS2 scan of the fragments, the B and Y ions, that make up that peptide. In a parallel track, we're going to go back to our genome database, identify all the proteins that are present in the genome. Since we know the genome sequence, we also know the amino acid sequence. In silico, using a computer, we will digest all the proteins into peptides that end again in lysine and arginine residues. And then we're going to calculate theoretical spectra for every single peptide in the genome. Let's say, for the sake of argument, there are 20,000 genes in the human genome, and each one of those has 10 tryptic peptides. The numbers are larger, but let's just say that for argument. So 20,000 genes times 10 tryptic peptides means there's 200,000 theoretical spectra, which we have to compare with an experimental spectrum. And again, we generate thousands of these during an experiment, so you can see that computational speed will be a consideration for this. But now at least the outlines of the computation are clear. The goal is to see which of these 200,000 spectra match the experimental spectra the best, and if so, is it good enough to be considered likely to be real? That is it the match above a certain threshold? And a very simple way of calculating the similarity between these theoretical and real experimental spectra is to simply perform a correlation between them. On top of that, mass spectrometrists have put all sorts of heuristics into the correlation calculation, like, is it good enough? Are there enough amino acids in a row that match up? But the fundamental calculation here is a correlation between 200,000 theoretical spectra and each obtained experimental spectrum. So after doing that calculation for all 10,000 or so experimental spectra, we get a series of matches. And the first thing we're going to do is we're going to organize the results by protein. Again, we're analyzing peptides, small protein fragments in the mass spectrometer, but we're mostly interested at the protein level. And here we see that the results are being tabulated and saying that it seems that we have good evidence for the presence of a protein called peroxyredoxin 1. And beneath that, we see that we have observed 6 peptides with a high confidence match to theoretical spectra. And if you observe 6 peptides of good quality, that's overwhelming evidence that the protein that you think is present in your sample actually is. A few other details that also will report the particular scan in which is being matched to the peptides. Here's some of the statistics. Here's the next, how much better the first match is than the second one. And then I'll say here's the number of ions that are the B and Y ions that are matched in the spectrum. Overall, for the protein, you see that we've identified 30% of the sequence, which is great, that there were 6 peptides, which we observed a total of 13 times, that this protein has 199 amino acids and a molecular weight of 22,000 or 22 kilodaltons. So all of those assignments that we made on the previous page were done with the assumption that there are no post-translational modifications. That is, we computed the theoretical spectra based on the presumed molecular weight of each amino acid. But many amino acids have post-translational modifications, and that's going to change their mass. I've listed here at the bottom 3 of the common post-translational modifications and how much it would change the mass of the amino acid. In this case, I have lysine. I've showed the difference between a lysine and a lysine that's been acetylated. You will recall that many lysines are acetylated in the histone tail region of those proteins, and this acetylation event modulates the interaction of histones with the DNA based on electrostatic interactions, and that this is a way of regulating gene expression, turning genes on or off. Acetylation adds 42 daltons to the lysine molecular weight, which I would guess is around 120 daltons. In essence, the way that you identify post-translational modifications are by shifts in the spacing between successive peaks. The way that one does this calculation is one computes all the theoretical spectra for normal lysines, identifies the best matches, and then calculates it a second time with lysines being acetylated. So by considering one post-translational modification, we've essentially doubled the amount of computation we need to perform. Whichever match is best in the cross-correlation is the one that we would assign. But still, computing power such as it is, we generally are parsimonious with our exploration of post-translational modifications because it does increase the computation time exponentially, and also it works as a bit of a fudge factor because what we're saying is, well, there are more ways to match the proteins, the peptides, to the spectra, right? You could say lysines can be this heavy or this heavy, and so you're just adding more variables, and of course, if you're not careful, you will get more matches because you have more chances of matching. So this has to be guarded against, and this is where a little bit of experience can help in the interpretation of these spectra. But again, fundamentally, the way we identify post-translational modifications is by observing increased masses in amino acid residues, and we ask the computer to consider those post-translational modifications which we think are most likely to be present at some time. Now, in talking about protein quantification, I want to revisit mRNA quantification, which we did with DNA chips. What you'll remember there is that we took 2 RNA samples, we labeled them red and green, we hybridized them to a chip, we calculated their fluorescence independent levels, and then we calculated ratios or observed ratios. If we observe a red spot, that indicates the test sample was greater than the control. If we see a green spot, we think the control levels are higher. That means the test sample has gone down. If we observe a yellow spot, that means they're roughly equally present. And if we see a black spot, that means they are equally absent. To measure 2 protein samples, we use the same concepts, but since we are not measuring fluorescence, we don't look at colors. What we instead do is we take proteins and we label them chemically, and since we're weighing them, essentially by mass spec, we're going to add light linkers or heavy linkers. These linkers are as identical in chemical composition as they can be, and then we're going to mix the samples together. They will elute at the same time since chemically they're very similar, but they will have different masses, so they will appear at different places on the M/Z spectrum, and by looking at the relative intensities, we get an approximation of their relative abundances in our protein samples. This technique is called ICAT, which stands for isotope coded affinity tag, and that refers to that the difference between the 2 linkers are based on isotopes. It can be of hydrogen or carbon. This example shows regular hydrogen and deuterium, and although there's some issues, that works out pretty well. So again, in comparison to mRNA, we're calculating ratios of intensities at different masses, and then we can compute the relative abundances of proteins in two samples. So now we'd like to end with a discussion of the comparisons of protein and mRNA abundances. But to start, I'd like to show a pretty cool fact about how you can calculate protein abundances with some degree of accuracy just based on the genome sequence. And here we have a property of a coding genome sequence and protein versus protein abundances, and you can see we have a reasonable correlation here. That if we calculate this property called the codon adaptation index, we can get to a good approximation a protein abundance. There are obviously some errors here, but this is only an approximation. So I'd like to remind you that with 61 amino acid coding codons, 3 of them were stop codons, as you recall, so there's 61 different ways to code for an amino acid for 20 amino acids, so that means there's a redundancy here. Here we see 4 different ways to code for alanine. Now what we've learned is these 4 different ways of coding for alanine are not equivalent, and that over time evolution has helped organisms and cells select that codon which is most appropriate for that protein. For the sake of argument, let's say that this is the codon used in the most abundant protein and this is the codon used in the rarest or least abundant proteins. That would imply that this one is going to get used much more frequently, and so you might expect that there are higher levels of that tRNA so that it could be incorporated more quickly into a growing polypeptide, allowing the cell to make more of that abundant protein. So I think that's pretty neat, and one of the really interesting aspects of this is it shows you that, in principle, if we knew enough about how to interpret the genome, we could, in principle, even identify how much of different proteins are being made at different conditions. It's an exciting prospect, and we can speculate about this topic a little more in class. So actually getting on to the comparison with mRNA, this work is basically based on 2 papers that appeared around the same time, around 1999. And what they basically show is that, as depicted here, it looks like mRNA levels and protein levels are pretty highly correlated. High mRNA levels correspond to high protein levels. And indeed, probably 90% of all papers published that compare protein and mRNA levels say, yes, we have high correlation between the two. But these original authors did a very insightful analysis and actually observed something quite different. The punchline is that you have to be aware of correlation coefficients because they're dominated by outliers. They are dominated by the largest values. There are only a few points here, maybe 6 or 7, where there are hundreds of points right in here. And so these 100 points are not having nearly as similar an impact as these 7 are here. And here I've shown a blow up a little bit of this area down in here, and here you could draw, based on these, a very different line at a very different slope. But because these 7 peaks happen to be where they are, we've drawn a very particular line, and to the eye that looks quite reasonable. But again, I want to warn you that correlation coefficients are dominated by extreme values. Now let's look at what they did when they started calculating the correlation values. They were correlating the amounts of mRNA and protein starting at those that are expressed at the lowest levels. And what they saw was that the correlation coefficient for the first 85% of mRNAs and proteins, there's almost no correlation. As you know, correlation coefficients can go from 1, very correlated, to -1, anti-correlated, and no correlation is a value about 0. 0.1 is extremely low, and people would say that's essentially no correlation. So for 85% of the proteins or genes included in the cell, there's essentially no correlation between the RNA levels and the protein levels. It's only when you start calculating at this very high end of abundances that correlation coefficients can get quite high. And although the number's blocked off, this is about 0.7 or 0.8. And that's what you see reported in the literature. Correlation coefficients of 0.8, which of course causes the reader to think, oh, protein levels and mRNA levels are very similar. But it's a very misleading number. Most protein and mRNA are not closely correlated, even though for the most abundant proteins they are. And again, it's biologically sensible. There's a mechanism. If you have a very highly expressed protein, well, you have to make about as much mRNA of that gene as you can, and then you have to make about as much protein from each of those RNAs. And so it is mechanistically reasonable that at very high levels they would be correlated. So the last thing I want to say about proteomics is just a discussion about one of the limitations of proteomics, and that is that unlike nucleic acid methods where you can easily amplify small samples, there's no simple way to amplify proteins. There are some ways, but very complicated. There's nothing like PCR, for instance, for proteins. So mass spec is a method that relies on basic abundance, and most all commercially available mass spectrometers are sensitive enough to detect about a femtomole of a protein. Again, femto means 10 to the minus 15th, and that sounds very sensitive, 10 to the minus 15th moles, but that is a large number of molecules, 10 to the 9th molecules. So think if we were trying to identify protein abundance in a small sample, maybe 1,000 cells. To get to 10 to the 9th molecules from 1,000 cells, that means each cell would have to have 10 to the 6th copies per cell. 10 to the 6th copies of a protein. Well, there are not very many proteins that are that abundant in a particular cell. There are a few, actin, tubulin, again, some of these very abundant proteins, but most of the proteins we're most interested in are not nearly at that level. You have to get up to quite a number of cells, let's say 10 to the 7th, before you can detect proteins that are relatively rare, maybe 100 copies per cell. And that's just a fundamental limit to the detection of a peptide, which you have a sensitivity of a femtomole. There are methods of detecting proteins that are more sensitive for individual proteins, like Western blots, which use antibodies, but that's a measurement of one protein at a time. So definitely people would like to develop new technologies that were capable of assessing thousands of proteins on small samples with only 1,000 cells. Again, we're able to do that at the mRNA level because we can take the mRNAs from 1,000 cells, amplify them using RT-PCR or other methods, and then put them on a chip or sequence them using next generation sequencing. Okay, thank you very much for listening.